Sorry about that. Yeah, so I'm a software developer. I lead a small development team in Seattle. This is going to be a very software-centric talk, I guess, but I'm really happy to have a chance to talk to so many people generating Mave data and also using Mave data for various purposes. I'm going to try to give a quick overview of MaveDB, but then really talk about what's happened recently in it and some of our roadmap going forward.
Okay.
Okay.
Go ahead. Keep going.
Thanks. So we've been hearing about the volume of data produced by multiplex assays of variant effect that has grown tremendously over the past decade. And according to this figure, sorry, having problems with the technology here. According to this figure, there were about 11 million variants analyzed by 2022. So this raises a problem of data collection, aggregation, and dissemination. That's where MaveDB came in. There we go. That's where MaveDB came in. It was launched in 2019 in a project led by Doug Fowler, Fritz Roth, and Alan Rubin. And today, it contains approximately 2,500 data sets and 7.1 million variant measurements. If you haven't had a look at it, I'd welcome you to visit the website at mavedb.org. Hello? There we go. Just to give a quick kind of capsule view of what's in the database today, the largest part of the data are in human cells. Of course, many of the other model systems have implications for human biology as well. And 7 of the ACMG secondary findings genes are covered in MaveDB. So for the first part of the talk, I really want to just give an overview of MaveDB from a data aggregation perspective. Then we'll go on and talk about recent developments and dissemination. This is what a typical MaveDB score set looks like. On the left, we have 2 columns that are required. One gives the variant name or variant HDVS string. MaveDB does accept protein variant strings, nucleotides, and splice site mutations. And then there's a score column, right? The rest of this is all optional. And this will typically include things like counts, standard deviations, other statistics, and really anything else that the investigator thinks is relevant to interpreting the variant. So that's a really simple data model. And I guess as motivation for that, consider the heterogeneity of Mave assays. I've just listed 2 ways that they're heterogeneous here. There are, of course, many others, but to begin with, they differ in the kinds of system they're using, and they differ in the kinds of readout that they're using. So on the readout side, some assays produce counts, some produce intensities or measures of protein activity. And MaveDB's solution to this is to be not very prescriptive. However, kind of looking forward, we do want to be able to disseminate data in useful ways to a variety of users and to support a variety of use cases. Those include clinical users who will be interested in just coming to MaveDB for information about one variant, machine learning users who might want to look at the whole MaveDB data set and perhaps work on predictive modeling, and users who really just want data to be available in other data sources like clinical databases. I want to remind us of what MaveDB's data sets look like and how they're organized. So on the right here, there we go. On the right here, we have an icon representing the score set we just saw. When you upload a score set to MaveDB, you'll be asked for some metadata that includes the analytic methods used in the kind of post-sequencing step, all the data analysis. And then the score set that you upload is part of an experiment, and so you'll also be asked to upload methods for the experiment over here. An experiment can have multiple score sets, and then experiments are also grouped into experiment sets, which might represent entire studies. One motivation for this structure is that there are complex experimental designs. In this example here, we have two paths, right? So this is a deep mutational scan of the BRCA1 ring domain. On the top, the top path here, there was an autoubiquitination experiment. On the bottom half, we have a yeast-2 hybrid assay. So each of these experiments then produced 2 score sets. Another thing that the data model is designed for is reanalysis. So MaveDB has a meta-analysis capability where you can choose a number of existing score sets and upload your reanalysis of them. So this is intended mainly for either imputation of missing values in a single score set or statistical reanalysis of multiple score sets. Okay, so I want to talk a little bit about our roadmap. What we've covered just now describes MaveDB as it was last year. It was launched in 2019. In 2022, MaveDB 2.0 came out. That added meta-analyses. It added the MaveHDVS standard for variant representation. And it introduced a REST API that you can use to integrate with your own software. At that point, there were about 3.6 million variants covered. So from there, we'll move on to recent work. And I'll show some screenshots of what we have today. The recent work is really grouped into 5 categories here: website enhancements, features for dataset contributors, platform improvements, features for data users, and preparation for clinical features that we want to introduce later. I'll walk through some of these. So to begin with, if you visit MaveDB today and you haven't in a while, you'll notice some new stuff on the homepage. These are just ways of allowing people to explore the data set. There are score set highlights here. You can look at kind of the top organisms, the top genes. You can look at DOIs and publications and keywords. So if you wanted to find all of the, I don't know, saturation genome editing data sets, you could look under keywords here. And find a link to them. Another way of exploring the data is to search by publication. So this was kind of a glaring omission earlier. We've added a more detailed publication data model. You can now search by author or title. You can, as a score set contributor, you can assign primary and secondary publications to your data set. And you can also associate preprints with your data set. So in this example, I've searched for Doug Fowler and found a whole list of data sets associated with his publications. We've zoomed in here on one of them. And if we were to click through this, we would get a list of other score sets based on this publication, and we could link back to the original publication. I want to talk a little bit about the improvements we've made in the data intake process. Really, kind of the 2 themes today are improvements in the contribution side of things, and then work that we're doing to support dissemination. So on the contributor side of things, if you were to visit MaveDB today, you'll notice a few changes. I'm going to walk us through the screens for creating a new score set. So first you log in, and if you haven't logged in a while, you'll notice that process has improved a bit. Some of the frustrations should be gone now. The first thing you need to do is create a new experiment. And you'll be prompted for a title, which will appear on the experiment page, a short description that'll appear in search results, keywords, DOIs, publications, and then your abstract and methods. And again, for the experiment, the methods are intended to really cover everything up through sequencing. So here I filled those out. After I submit this, I'll create a new score set. And here's where the screens have changed a little bit. This is sort of very recent changes. So we've taken the kind of complicated form for data intake and broken it into a series of steps that are guided. We'll be doing more of that, so you should see more changes happening over the next few weeks. To begin with, we're prompted about whether this is a a score set that corrects or supersedes another data set, or whether it's a meta-analysis of existing data sets. Given that it's not, we're asked to associate it with a new experiment. On the next screen, we're asked for the usual kind of metadata. So again, a title that'll appear on the page, a short description for search results, and an abstract. The abstract is pre-populated. Similarly, keywords and publications are pre-populated, but we can edit them as necessary. And one thing I want to draw attention to is the license field here. We recommend using Creative Commons 0 for everything. We're able to then publish those to Zenodo, but a variety of other licenses are available. You can also associate your own custom data use terms. The next step is to assign targets to your score set. So in this example, I've already entered a target gene name, I guess, yeah. And we're going to upload a FASTA file that gives the sequence that we're working with. It's useful to assign a uniprot ID here and say what the offset is of the sequence that I'm working with. And that's all that MaveDB really requires. If you're doing endogenous genome editing, like saturation genome editing or prime editing or base editing, then you'll want to provide the target in a different way. So we now support genomic coordinates. You are asked to choose an assembly, enter a name using the HGNC nomenclature, and then you can choose an accession number or transcript. But really, again, that's only for endogenous genome editing. And the last step is to upload your scores and your counts. So having done that, what do you get? We've wanted to add features that would give people some value right out of the box. More of that is coming, but to begin with, we have a score distribution, and we have a heat map of variant effect. One thing to note about the heat map right now is that we don't currently have any information about how the investigators interpret their own scores. So the range for the heat map is the same for everything right now. That's something we intend to address shortly. In addition, there's a section of curation metadata. It lists the license terms. Users can download your scores. We have key text elements like abstracts and methods. The references you've supplied, target information, and a preview of the data. A few things we've added to the screen lately include the ability to search for a variant. Here you can enter all or part of an HDVS string and find the variant. It'll show you the score and where it lies in the distribution. If it's a single amino acid mutation, it will also show up on the heat map. A couple of last things for contributors. The data validation is more robust now and also more verbose. So if there's some kind of validation error or inconsistency that MaveDB can't handle, it will be pretty explicit about where that lies in your data set. Next, for those of you who might be submitting high volume data, doing that through the web interface isn't necessarily convenient. We have a pretty extensive REST API that you can use. This screen here is from the documentation, which you can find linked to the MaveDB website. You can run sample queries and things right here in the documentation. There are about 70 API methods at the moment. And so to support that, we have really 4 Python libraries, or well, four code repositories. The top one is the main MaveDB library. That includes the things like the data model, our validation code, also the API server. The web interface is a separate application. And then as score set contributors, you might be interested in the MaveHDVS library, which is a reference implementation of the MaveHDVS standard, which is a subset of HDVS nomenclature. And finally, MaveTools is a very lightweight client library for interacting with the API, whether you're fetching data or contributing data sets. Since I'm a software developer, I couldn't resist putting in one architecture diagram here, just to show how the various pieces interact. We've rewritten the application using contemporary open source components. There is kind of the main module here, which is the MaveDB API. When you submit data to it, asynchronous processing happens in another server that will validate your data set. Pretty soon, it will also map your variants to the human genome, if appropriate. Over on the right-hand side, we have the web interface. And at the top is a client application that could be a piece of software you've written. It could be a Jupyter notebook. Which brings me to this slide. Alan Rubin has written a nice example of a Jupyter notebook for submitting data sets. And I think it's not yet in any of our code repositories, but it will be shortly. So if, in the meantime, if you're interested in looking at it or using it, just contact me. One other thing that's happened recently is we've submitted a complete data set of all of the Creative Commons Zero licensed data to Zenodo. So this might be of interest to people doing predictive modeling of variant effect. There's it's about 1.4 gigabytes in size. There's a JSON file that describes all of the experiment sets, experiments, and score sets. And then they all have their own individual CSV files of scores and counts. Other recent work, I've alluded to the new login mechanism. The only other thing I'll say about this is that we've done a lot of work behind the scenes to improve the stability of the application, maintainability, validation, and so forth. Finally, I want to talk a little bit about the roadmap for the rest of this year. We were here. During this period, since last summer, we've expanded the team size quite a bit. There are now 5 developers, 2 of them working full-time on this. We've hired a lead developer who's devoted to this project full-time as well. And in the interim, the data set has grown to, this is actually out of date, it's now 2,500 score sets and 7.1 million variants. So where do we go next? The work we are undertaking right now is really focused, on the one hand, on features for contributors, and on the other hand, in preparing to release features for clinical users. So I've talked a little bit about the beginning of the data intake interface improvements. I won't say too much more about that. But I want to talk about some of the challenges of disseminating data. So looking back at the variant search screen that I showed, you can search for a variant within a score set. That's probably not useful to clinical users at this point. It's important to be able to do it, but we can't search for a variant of a gene in the human genome, or we can't search for the gene itself and see all of the score sets that relate to it. In order to do that, we're going to need to be able to map variants to the genome. And so we've been working with the Wagner lab. They've written a very nice utility that does this mapping. And we're working to integrate it into MaveDB. So it takes as input our variant here. It generates a verse representation of that variant in the model system. So a verse is a more verbose, but more computationally useful, to us anyway, representation of the variant. And then it applies a sequence ontology to map that to the human genome and to generate a verse representation of the mapped variant here. What that will get us is the ability to add screens to MaveDB that describe variants in the human genome. So here's a mock-up of what we intend to create. The first thing to notice here is this chart, where we have the distribution of variants in a score set. We have functional and non-functional ranges shown according to the investigator. And then we also have variants in that score set colored according to their known clinical status. So here we see pretty good concordance between, at least to me it looks like pretty good concordance between the clinically known variant effect and the prediction. Once we're able to do that, we can generate odds paths where appropriate, and clinical evidence codes perhaps, and provide links to other data sources and variant-specific information here at the bottom. So this is really the major goal that we're working toward right now. As I said, one thing we need for this is the ability to map things to the human genome. But we need some other information too. We don't know at the moment how an investigator interprets their score ranges. That's why I said the heat maps are scaled uniformly for everybody at the moment. So we need to ask that question. And our intended approach to that is to ask what score ranges the investigator used and how they might map to a standard ontology. Another thing we'd like to be able to display is information about things like what types of variant the experiment can detect. So in short, additional contextual information that would help clinical users to interpret the data. To that end, the AVE-DCD workstream has produced a set of minimum information standards. We're working to implement those in MaveDB. And at the same time, we have a team in Seattle working to curate existing data sets. They're going back to the original papers and building a set of metadata. So we'll use their recommendations as well as we extend MaveDB. So I've talked about mapping to the human genome, about generating clinical pages for each gene, and about metadata curation a little bit. There are plenty of other things we're interested in doing. We want to add metadata for data-specific columns so that if you have custom columns that you think are relevant to anybody using your data set, we want to be able to describe those. We'll also make recommendations about standard columns like standard deviation. We'd like to pursue some kind of some kind of integration with Mave Registry. What that looks like exactly, I'm not quite sure yet. And I'm interested in hearing from people about what would be most useful, right? And finally, a big goal is to get MaveDB data into clinical databases. So that's something we're beginning to work on, and we'll certainly continue through the remainder of the year and into next. So I'm really interested in hearing from people over the next few days. After that, we have a Slack channel in the AVE Slack workspace. If you're a dataset contributor, please message me if you'd like to be added. We try to respond pretty quickly there to any issues you may be having. We also have a new Google group that doesn't have much in it yet, and you're all welcome to join that. And that's the end of my talk. I'd like to acknowledge many people who have contributed to MaveDB, in particular our development team in Seattle and in Melbourne, the MaveDB investigators, other coworkers at the Broughton and Beatty Institute, past MaveDB developers, members of the AVE Alliance, including the Wagner Lab, and also Erwan Delage, who's worked with us a bit on to help us ingest saturation genome editing data. Thanks.
Thanks, Jeremy. Very interesting. To start, I've got two kind of related questions. One is there any push to make it a requirement of journals publishing Mave datasets to submit in MaveDB like it is for lots of other kinds of data? And also sort of related to that, is there a mechanism currently to submit and get an accession number but not make it public right away? Because I've encountered that where, as a reviewer, I suggested submitting to MaveDB and the author said we'll submit after because we don't want to make it public yet.
Right. So to answer the second question first, yes, certainly you can submit data to MaveDB. You can generate a temporary URN, not publish your data set yet. As we add visualizations and things that you might find useful for papers, you'll be able to generate those and export them without yet having made your data set public. So yes, for sure. To your first question, though, is one I'm probably not the best person to answer. I think that might be a question that some of the other MaveDB investigators could answer better than I can. So I might defer to Alan Rubin and Doug Fowler on that one.
I have a mic. So it's increasingly becoming possible to make many, many, many measurements at, say, genome scale. So how do you envision MaveDB interacting with high-dimensional data sources, single-cell RNA-seq, imaging, et cetera?
Yeah, that's a really good question. I would love to talk about it more. I don't have I don't have a data model prepared for that yet, but we are definitely thinking about it already. I guess I would like to understand what seems to make the most sense from the standpoint of those who are generating the data set. But I'm glad to hear the question.
So I'll just answer the question about journals. I think that it's premature for us to expect that the journals and the journal editors are going to do anything. But I think that it's really something that those of us who are engaged in the community, as you review papers, if you ask that, or if you say that it's a requirement that the data be shared in a repository like MaveDB, if we as reviewers ask for that consistently, I think that's how we would get a sort of adoption by the journals. The conversion rate, in my experience, is 100%. In terms of getting getting data sets added to MaveDB as part of review.
People that they have to.
I do. So I think that if more people do that, then that will work. And I think that we are, as people who are involved in the MaveDB project, we're always very happy to provide support to individuals who want to upload their data. We know that, as Jeremy said very, very nicely, I mean, all the data sets are different. Sometimes it's hard to figure out how your experiment fits into the data model. But so we're always happy to engage.
Just repeat that for.
Yeah, so the question is about if an investigator wants to provide a set of controls instead of relying on the data in ClinGen and ClinVar, can they upload that with their data set? There's no mechanism for that yet. In fact, the screen that I was showing is a mock-up at this point, but we do expect to be getting that out in this year. I think the feature you're describing makes a lot of sense. I'd want to talk to others about it and kind of get everybody's take on how it would best work. I don't see any problem doing it, though. Yeah.
I can speak loudly anyways. I would just counter that with what is the data you're going to provide to say your controls are benign or pathogenic, right? That's the situation we're in right now. So it's great if it's done and people can support it. But so I'm just saying I'm countering the ask, can you bring your controls? Great, but I think if you're going to bring your own controls that are not in ClinVar or in other places, that you would also need to provide the data of the evaluation of that for others to use. Otherwise, we're in the same sticky place, right, that we're in right now.
Are you doing any work or looking at visualizing pairwise mutations? I know that you have this really great viewer for single mutations.
Right. I've been thinking about that a lot. And I mean, I have some ideas about how we might visualize them. I'm not sure what's most useful. And I would love to talk about that more over the next couple of days if you have any time. But it's definitely been on my mind for a while. And other things we'd like to do are also compare score sets, right? Which is something I didn't show here.
Thanks for a great talk. I have a
kind of more abstract question, I guess, about the future of the database, which is a wonderful tool. So obviously the effect of these variants is often extremely contextual based on the experimental system. So I guess I wanted to kind of ask you about your broader thoughts about building these kinds of coherent data models at the level of a locus when you have potentially highly conflicting data across different experimental systems. And that's even before getting into things like different isoform expression and things like that.
Yeah, I think generally we don't envision MaveDB as a tool that has a high degree of curation. So we want it to be a place where everybody can deposit their data. But then there's a balancing act because we also want to make that data available and disseminated in some kind of usable form. That's a balance that's still being struck, I think. I'm not sure if that's a very complete answer to your question, but it's definitely the challenge that we've been working through.
I guess I just want to say I celebrate your ambition to let the bear dashboard, but that also sounds very hard.
Yeah, I think we can provide some contextual information that'll help. For instance, if somebody searches for a gene and we have a number of score sets that relate to it, we can show the distribution of scores in those score sets. Contextual information like that might help in evaluating these things. Again, I don't think it's probably MaveDB's role to go much further than that. We want to provide, investigators with the ability to generate figures and things that they might use, and we want to provide a place then where we can aggregate this data and export it to ClinVar and ClinGen. I'm not sure how tractable the problem would be to really try to curate that data heavily in MaveDB.
Well, if that's everything, let's thank Jeremy for a very nice talk.